Honorable Chatpalli sir, who gave us a wonderful insight right from the historical background of North Karnataka to the present day potential of North Karnataka to grow in various sectors from agriculture to arts and culture. It was a wonderful lecture, sir. Thank you very much. ಇಲ್ಲಿ ಆಗಮಿಸಿದ ಹಿರಿಯರಾದ ಶ್ರೀ ದರ್ಜಿ ನಾಡಗೀರವರೇ, ಮಿತ್ರರಾದ ರಘುಮಂಚಿಯವರೇ, ಅಕ್ಮಂಚಿಯವರೇ, ನಾಗೂರವರೇ, ನಾಯ್ಕ್ ಅವರೇ ಮತ್ತು ಇಲ್ಲಿ ಆಗಮಿಸಿದ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರೇ, ದಿಸ್ ಲೆಕ್ಚರ್ ಈಸ್ ಎ ಲಿಟಲ್ deviation from the socio-economic developmental aspects of North Karnataka, and though it is concerned with North Karnataka, Director CMDR day before yesterday asked me to come to this session. I am actually an alternative for one chairman of KVG who was to come here to chair the session. Then I said, with regard to North Karnataka, we have also done a lot of work in a different aspect that I would be pleased to share the work that we have done and the issues related to an entirely relevant aspect of North Karnataka, the health issues of the people of North Karnataka. In the next 15-20 minutes which has been given to me, I will be talking on the impact of cultural practices on the health conditions of North Karnataka population. Now you may be wondering how the cultural practices, traditional cultural practices, some of them, not all, have their impact on the health of the people of any population for that matter. We have done some work on North Karnataka population in this institute, which is called as Karnataka Institute for DNA Research. This is the first of its kind in the entire Karnataka, established in 2010. I am the founder director of this institute. Then I became the Vice Chancellor of Karnataka University. Presently now, after stepping down from the Vice Chancellorship, I am again the Director of Karnataka Institute for DNA Research. This an institute was established actually looking at the cultural practices which have their impact on North Karnataka population. Way back 3 decades ago, I was teaching human genetics. I have been teaching human genetics for more than 4 decades. I took up a project called as consanguinity and mental retardation. Consanguinity is the marriage between close relatives, which is found in high frequency in Karnataka, especially in rural North Karnataka. This project, I did it in Hubli, Dharwad and other places where mentally retarded schools are there. I interviewed the parents of the mentally retarded children, where I came to know that staggering statistics, 72% of the parents of the mentally retarded children were relatives. Relatives in one way or the other. So that means these marriages in relatives, blood relatives, result in certain abnormalities in the children. Then I thought of establishing an institute. I submitted a proposal to Government of Karnataka, made presentation, and way back in 2007-8, I got an individual research funding of 3 crores for the first time in the history of Karnataka University, with individual principal investigator and 3-4 co-investigators. With that, this building came. In 2013, it was converted into an autonomous institute called Karnataka Institute for DNA Research. The primary objective of this is to identify the genetic disorders and cancers which are prevalent in high frequency in early stage so that they can be prevented. So in this lecture, I will just give you a brief idea about what are the cultural practices which are being practiced in not only North Karnataka, in entire South India. Karnataka, Andhra Pradesh, Tamil Nadu, Kerala. When compared to North India, the frequency of consanguineous marriages is very high, and thereby the coefficient of inbreeding is also very high. Hence we have highest frequency of congenital malformations and genetic disorders and even cancers. Figures of the laboratories which are there in Karnataka Institute for DNA Research. We have 4-5 laboratories with state-of-the-art equipments to conduct research on human molecular genetics. So these are the 4 laboratories. This is microarray core facility where we can analyze thousands of genes at a time to identify the different types of genes be responsible for different diseases. So this field, human genetics, is a very fascinating field with tremendous social relevance. It directly is responsible for the health of the individuals. Genetics is the science of inheritance, and inheritance is the transmission of characters from parents to children. We have seen that we inherit our characters from our parents, grandparents, great-grandparents, etc. When a child is born in a family, all the relatives gather around the child and try to see the similarity of the child features with their ancestors. Some may say that the eyes are like the mother, or the forehead is like the father, or the ears of the child are like grandmother, like this. Why? Because the child inherits all these characters from its ancestors, not one generation, many generations together. The science of genetics, I'll be little fast because time is running, has tried to answer 3 important problems. One is, what is inherited? What is the physical and chemical nature of the hereditary material? This problem has been solved way back in 1953. 2 scientists, Watson and Crick, you must have heard their names, who worked in a laboratory in University of Cambridge, where I visited. Nagur knows, to sign an MOU when I was Vice Chancellor. We visited this laboratory where Watson and Crick worked and discovered the structure of the DNA, structure of the hereditary material. This laboratory, Cavendish Laboratory, you may wonder, a small laboratory has produced 29 Nobel laureates, and among them were Watson and Crick. So they identified the physical structure and the chemical structure of the hereditary material that we call as gene or deoxyribonucleic acid with the help of X-ray crystallography. The second problem is how the hereditary material is transmitted from one generation to the next generation. Way back in 1866, you must have heard the name of Gregor Mendel. Gregor Mendel conducted experiments on pea plants in the monastery where he was working, and he came out with the laws which are called as Mendel's laws of heredity. That means the hereditary material follows certain rules when it is transmitted from parents to children, from one generation to next generation. Though he conducted experiments on pea plants, then unfortunately for 35 years his laws were in darkness. Only in the turn of the century, 1901, that Mendel's laws came to light. Then the scientists tried to apply the Mendel's laws to smaller animals like rats and guinea pigs, and found that the laws were applicable to animals also. Then they wanted to see whether these laws are applicable to human beings. It is not possible to conduct experiments on human beings as it is done for animals. So we have a platform called as society, and in society we have families where one person is married to another, male married to female, and children are born. This is called as family. By analyzing the families, they tried to understand how the characters are transmitted from generation to generation. We have 2-generation families, 3-generation families. So with the help of this family analysis, which is called as pedigree analysis, they came to the conclusion that many characters, almost all the characters, are transmitted from generation to generation in Mendelian fashion. It was in 1909, a physician, English physician, Sir Archibald Garrett, wrote a classic book, Inborn Errors of Metabolism. In the Inborn Errors of Metabolism, he, with the help of his daily practice with the families, with the patients, he came out with the application of Mendelian principles in human beings in diseases. He worked on 4 diseases: phenylketonuria, alkaptonuria, galactosemia, and another one, I don't remember. The 4 diseases where he showed that these diseases, albinism, these diseases are transmitted from generation to generation in a Mendelian fashion. This was the first book on human genetics. What I am trying to tell is that today we know how the characters are transmitted from generation to generation. The third problem is very, very difficult. Still research is going on. How the genes act. The genes are present in human, I'll show you, present in the cell, in the nucleus, in the chromosomes. How do they act? They behave in a different manner in different individuals. So every individual is different from each other. That is the beauty of genetics. Beauty of the genes. The genes are different in different individuals, even though they are born for the same parents. Same parents, they give birth to 5 children. All the 5 children resemble the parents, but all of them are different in one way or the other. Not only in physical characteristics, but also in psychological characteristics. This is because of the combination of the genes that they get from their parents when the conception takes place. So this is the diagram of the cell. Those who are from social science, they can know cell. The central portion is the nucleus. You can see here. This is the nucleus, and inside the nucleus you have thread-like structures which are called as chromosomes. There are 46 chromosomes in human beings, and the number is fixed for every species. If the number increases to 47, the child becomes abnormal. If the number is reduced to 45, the child becomes abnormal. Inside the chromosome there are structures called as the double helix DNA. This is the gene which was identified by Watson and Crick. This is called as Watson and Crick double helical model. How does it look like? It looks like 2 staircases, spiral staircases intertwined with each other. They contain the bases, adenine, guanine, thymine and cytosine, phosphate and sugar. This is the chemical structure of the DNA. So this is the hereditary material that is transmitted from parents to children. This is responsible not only for normal characters but also for all the diseases that we know today. Right from diabetes to blood pressure to heart attack to cancers, the genes are responsible. They are also responsible for psychological characters like schizophrenia or any other psychological character. The genetic disorders in human beings are classified into different categories. There are more than 6,000 genetic diseases in human beings which are found in a website called as OMIM, Online Mendelian Inheritance in Man. OMIM, if you go to this website, you find large number of genetic diseases. Whenever a new disease is identified, it is entered into this. These are all diseases which follow Mendelian principles. These diseases are classified into single gene disorders, which are caused because of the change or mutation in a single gene. Hemophilia, thalassemia are single gene disorders. Chromosomal disorders, the disorders which are caused because the change in the number of chromosome or structure of the chromosome. Why I am telling you all these? They are more in North Karnataka population. Then you have biochemical disorders like phenylketonuria, alkaptonuria. All these are because of biochemical defects, defects in digestion, defects in circulation, defects in excretion. These are biochemical disorders. Metabolic disorders. Then you have multifactorial disorders caused because of the effect of environment and genes like diabetes. Diabetes is not completely controlled by genes. It is also controlled by environment or lifestyle disorder. I told you consanguineous marriages or consanguinity. Consanguinity is a phenomena. Consanguineous marriages are the marriages between close relatives. The term consanguinity is from Latin word con means with, sang means blood. It's actually blood relatives. If you marry your own sister's daughter, it's a consanguineous marriage. If you marry your mother's brother's daughter, it's a consanguineous marriage. Different types of consanguineous marriages are there. Now what happens when a consanguineous marriage takes place? These are the different types of consanguineous marriages that we find in North Karnataka and even in different parts of the world also. For example, if a person marries, first one is paternal parallel first cousin marriage. Paternal parallel first cousin marriage. If a person marries his father's brother's daughter, it is found in some communities. It is called paternal parallel first cousin marriage. If a person marries mother's sister's daughter, it is maternal first cousin marriage. If a person marries father's sister's daughter, it is cross cousin marriage. If a person marries mother's brother's daughter, it is also a cross cousin marriage. These are all first cousins. Then the second line you have second cousin marriages, 2 generations past. If you marry your mother's sister's daughter, or father's brother's son's son, that is second cousin marriage. All these types of marriages are called as consanguineous marriages. Apart from this, in some of the families we find still highest consanguineous marriages, which are called as incestuous marriages. I will show you. When you look into the history of consanguineous marriages in different populations of the world, we find that more than 1.1 billion people living in different countries of the world practice different types of consanguineous marriages. These marriages are primarily practiced in royal families. For example, royal family of, so this is the global rate of consanguinity throughout the world. Different countries, different populations practice different types of consanguineous marriages, and the rate of consanguineous marriages are also shown here. For example, royal family of Egypt, the pharaohs. If you see the Google, you can find out pharaohs of Egypt, they married their own sisters, and even they also married their own daughters. There are examples shown here who have married their sisters, who have married their own daughters. Now what happened? 1 or 2 generations, the children were okay. Next, they found that the children born to such marriages, consanguineous, they are now considered as incest, I-N-C-E-S-T. Incest is a prohibited marriage. But they practiced this kind of a marriage because they wanted that their royal blood should not go out of their family. Their royal blood should not go out of their family. The children started suffering from a very debilitating disorder called as osteoporosis imperfecta. Osteoporosis imperfecta is a brittle bone disease. If you just touch the bone will break. These are the different types of osteoporosis imperfecta. Mild, moderate and severe types are there. These children are born to highest incestuous marriages, and later on, after a few generations, the pharaohs of Egypt disintegrated. Because such children were born to such kinds of incestuous marriages, consanguineous marriages. The Habsburg family, it was started with Roman Emperor Rudolf in 1273. They also married among close relatives. Then they suffered from a disorder which is very famously known as, you can Google, you can see Habsburg jaw. You can see the jaw. This has run in several generations. The difficulty in chewing, variety of disorders, the to talk, growth disorders, impotence, malformed tongue, hair loss, human back epilepsy, hunchback epilepsy, mental illness, short-lived. Ultimately, the dynasty came to an end. This was the disorder. The children were born with the consanguineous marriages. Then we have the example of royal family of England and royal family of Russia. Family of Great Britain and England. Queen Victoria, the Great Britain, carrier of hemophilia. Hemophilia, I will show you the figure. Hemophilia is a disease of bleeding of the blood. If a person meets with an injury and the blood starts bleeding, it continuously starts bleeding. Kusuma roga anta adike. Why? Because they lack a protein which is responsible for clotting. Hemophilia, there are factors, factor 8, factor 9. If they are absent, why they are absent? The gene responsible for these factors undergo mutation. Why they undergo mutation? Because they have married in their close relation. Queen Victoria was a carrier of hemophilia. She married, her granddaughter Alexandra was also a carrier of hemophilia. She married Crown Prince Nikolai from Romanov dynasty of Russia. She gave birth to Prince Alexei, who was himself hemophilic. In Karnataka, especially North Karnataka, we have a hemophilic foundation of Karnataka. There are more than 2,500 hemophilic patients. We have collaboration with them and we have done a lot of work on hemophilia. The ultimate objective is to find out a carrier. The hemophilia gene has been identified. It is present on the X chromosome. We receive our X chromosome from our mother and Y chromosome from the father. So if the mother is the carrier, if she transmits the X chromosome to his son, the son will be affected. Therefore, in hemophilia, more number of males are affected, females are carriers. If we identify the carriers early, we can see that the hemophilia is eradicated from the population. And we are trying to do that with the help of Hemophilia Foundation. Consanguinity and genetic diseases in North Karnataka. Many workers, names are given there, they have worked on consanguinity and genetic disorders in Karnataka, Andhra Pradesh and Tamil Nadu populations. It is generally observed that the disorders which are controlled by recessive gene, there are two types of genes. One, recessive genes, and the other dominant genes. Dominant genes express if they are present on one chromosome. Recessive genes express if they are present on both the chromosomes. We get our 46 chromosome, 23 from the mother, 23 from the father. If my mother is also carrier, father is also a carrier, if they marry and I am born, if I get both the genes, carrier genes in double dose, I get that disease. Usually, the recessive genes are responsible for such disorders. And therefore, when the marriages of close relatives takes place, the children born are defective in nature or affected by these disorders, including cancers, including many of the cancers. One example I will show you. This is cystic fibrosis, which is found in many of the families. Cystic fibrosis is a disorder of the recessive gene wherein the child starts producing mucus in different organs of the body. It may be in the lungs, it may be in the kidney, it may be in the liver. When the mucus accumulates, it has to be removed. It's a continuous process. Why it happens? Because it is caused because of a defective gene responsible for cystic fibrosis. This gene is recessive in nature. Since it is recessive in nature, the mother are carriers, they are normal. And the child, when it gets these two genes from the parents, it has got double dose, it is homozygous condition. So that is the reason when consanguineous marriages take place, there will be homozygosity of the recessive genes. You can see here. The dark in the fourth generation, you can see the last child, 4-4 is affected. And that is born to 3-3 and 3-4. 3-3 and 3-4 are relatives. Why? 3-3 has married his mother's brother's daughter. Mother's brother's daughter. Both of them are carriers, half. When they marry, they produce one normal, two carriers and one affected. This is 1:2:1, as if you have seen Mendelian ratio of segregation. So this kind of disorders are found in marriages which happen between close relatives. You can see the child which is suffering from cystic fibrosis. This is the statewide representation of consanguineous marriage rates. You can imagine, you can see in North India, the frequency of consanguineous marriages is very, very low. In South India, the frequency of consanguineous marriages is very high. In Afghanistan, Pakistan, the consanguineous marriages frequency is very, very high. So in populations where consanguineous marriages are high, the frequency of genetic diseases and cancers is very high. So these are some of the disorders that we find. Hemophilia, I told you. This is a bleeding disorder which is born, with the child is born to parents who are close relatives, who are carriers of hemophilia. In Karnataka, large number of children are there suffering from hemophilia. The best way is to counseling, to bring awareness among the children, among the parents about this. This is another disease on which we are working, sickle cell anemia. Sickle cell anemia, as the name indicates, the red blood cells which are normal in normal individuals, they are oval in shape. In sickle cell anemia, the red blood cells become sickle shape, sickle andre kudugol. Why? Because of the defect in the hemoglobin structure. Hemoglobin is a red pigment present on the red blood cells. In normal hemoglobin structure, has got 4 chains, 2 alpha chains and 2 beta chains of amino acids. Chains are amino acid 1, 2, 3, 4, 5, 6. They are arranged in a particular order. 2 alpha chains and 2 beta chains are there in a molecule of hemoglobin, normal. What is the difference between normal and abnormal hemoglobin which produces sickle cell disease? In abnormal hemoglobin, both the alpha chains are same as normal hemoglobin. One of the beta chains is also same as normal hemoglobin. In the other beta chain, the 6th position, 1, 2, 3, 4, 5, 6, 6th position of amino acid, in the normal hemoglobin, it is glutamic acid. In the abnormal hemoglobin, it is valine. So glutamic acid is changed to valine. So this small change in the amino acid results in sickle cell anemia. The child wants blood to be blood transfusion every 15 days. So sickle cell anemia is a very, very debilitating disease for children. They may not survive for a long time. But fortunately what has happened is sickle cell trait, which is in a heterozygous condition. See, heterozygous condition is an advantage in malarial environment. So people will not suffer, sickle cell trait people will not suffer from malaria. So sickle cell gene is more found in malarial endemic environment. In South Africa, the frequency of sickle cell gene is 25%. In India also, in some of the tribal populations, the sickle cell gene frequency is high. This is Down syndrome, wherein the chromosome number is 47 instead of 46. You can see the 21st chromosome. This is karyotype, arrangement of chromosomes in the individual. First chromosome, 1 from father, 1 from mother. When we arrange in the laboratory, we find that 21st chromosome is not 2 in number, 3 in number. It is trisomy 21. The child becomes abnormal. This is similarly Edwards syndrome, wherein the chromosome 18 is 3 in number, trisomy 18. All these children are mentally retarded with different types of abnormalities. Most of them are born to consanguineous marriages. This is Patau syndrome, chromosome 13 is 3 in number. This is Turner syndrome, which is the defect of X chromosome, sex chromosome. The 23rd pair is X chromosome or Y chromosome. If it is XY, it is a male. If it is XX, it is a female. In this case, there is only one X chromosome, there is no Y chromosome. The child is female, but it is not a productive female. This is Klinefelter syndrome, where you find 2XXY, double XY, 47 chromosomes. You must have seen the eunuchs, who are, majority of them are of this karyotype, XXY 47. Then you have this Cry-Duchat syndrome, wherein the 5th chromosome, one of the chromosome is deleted, partial deletion is there. The child suffers from a syndrome called as Cry-Duchat or cat cry syndrome. It cries like a cat. So what I want to highlight, especially with respect to North Karnataka, since the frequency of consanguineous marriages is very high, the genetic disorders and cancers, we have worked on cancer, breast cancer, large number of breast cancers we have collected, families we have collected. The major point is to bring about awareness. And second point is to identify these disorders well in advance, because once the child is born with a genetic disease, it is difficult to cure. It will be a burden to the society, it will be a burden to the family also. So what has to be done? It is to be identified when it is growing in the womb of the mother. There are two important techniques. One is called as amniocentesis, where amniotic fluid is taken out and studied for abnormality at the 13th or 14th week of gestation. Another technique is chorion villi sampling, which is done around 10 to 12 weeks of gestation. Chorion cell is taken out and studied for chromosomal abnormality. If we find that there is an abnormality, there is a choice of aborting the child. So with the help of these two techniques, which we are standardizing, we are bringing about awareness. We are trying to, for early detection of these and also cancers, we provide service and as well as research. Karnataka Institute for DNA Research has done a lot of work in COVID in terms of identification with the help of RT-PCR, which we have been doing for the last 10-15 years. With this, I thank the CMDR for giving me this opportunity to share some of my work, especially for the people of North Karnataka. Thank you very much.
So for providing insights on various genetical disorders and its impact on the health conditions of the people, and how awareness is very much important for that, to avoid that. Now we shall take 2 or 3 questions from the delegates. Any questions? Please raise your hands.
With regards to regional imbalance, sir, good evening. I was also, while doing my PhD, was engaged in some of the projects which relation to the study of regional imbalance. So, sir, I mainly want to speak on how administratively the regions of North Karnataka are far away from Bangalore, and what is being done to bridge that gap. That is what I want to answer. The regions of North Karnataka and especially of Hyderabad, Karnataka are too far away from the administrative capital, that is Bangalore. And that is one of the reason that I think, and after studying many of the research papers, I found that those regions were quite far away from the administrative region, and that is why many of the regions were kind of neglected. For example, if we go to places like Bidar, and if we go to places like, what we can say, like Aland and other regions which are technically too far away from the region, from the capital of Karnataka, those places are administratively very far away. And they are quite close to capital cities of other states than capital city of our own state. And that is one of the reason why those places have been thoroughly neglected. And even today, we can't actually find the presence of our state machinery in those regions. So what actually the groundwork has been going on to address these shortcomings?
Small is beautiful, always. There has been historically reasons for underdevelopment in these 14 districts. Northern Karnataka, which is historic also. I have not gone for detail. There has been a discussion how administratively Mysore, Karnataka paid attention on education, health and other developmental activities, and how these areas of Kalyan, Karnataka which were there, which were under the repressive rule of Nizam, ಆ ಕಾರಣದಿಂದ ಆಗಿಲ್ಲ ಅನ್ನುವಂಥದ್ದಕ್ಕೂ ಹಿಸ್ಟಾರಿಕ್ ರೀಸನ್ಸ್ ಇದೆ. ಸೆಕೆಂಡ್ಲಿ, ಬಾಂಬೆ ಕರ್ನಾಟಕದ ಭಾಗ, ದಿಸ್ ಈಸ್ ಆಲ್ಸೋ ಫಾರ್ ಅವೇ ಫ್ರಮ್ ಬಾಂಬೆ, ಮುಂಬೈ. ಹೀಗಾಗಿ ದೇರ್ ವೇರ್ ಲಿಟಲ್ ಡೆವಲಪ್ಮೆಂಟಲ್, ವಾಟ್ ವಿ ಕ್ಯಾನ್ ಸೇ, ಲ್ಯಾಕ್ಯುನಾ, ಶಾರ್ಟ್ ಕಮಿಂಗ್ಸ್ ವಿಚ್ ವಿ ಹ್ಯಾವ್ ಟು ಎಕ್ಸೆಪ್ಟ್. ಆ್ಯಂಡ್ ದೀಸ್ ಶಾರ್ಟ್ ಕಮಿಂಗ್ಸ್, ಇಂಬ್ಯಾಲೆನ್ಸ್ ತೆಗೆಯುವಂಥ ಪ್ರಯತ್ನ ನಡೆದಿಲ್ಲ ಅಂತಿಲ್ಲ. ಈಗ ನೀವು ಬೀದರ್, ಗುಲ್ಬರ್ಗ, ಯಾದಗಿರಿನವ್ರಿಗೆ ಹೇಳಿದರೆ ಅವ್ರಿಗೆ ಹೈದರಾಬಾದ್ ಭಾಳ ಇಷ್ಟ. ಆ್ಯಂಡ್ ದೇ ಆರ್ ನಿಯರರ್ ಟು ಹೈದರಾಬಾದ್, ನಮಸ್ಕಾರ, ದ್ಯಾನ್ ಬ್ಯಾಂಗ್ಳೂರ್. Those who are in Belgaum and Hubli, Dharwad, they feel they are nearer to Pune and Mumbai. This is a cultural aspect also. So there has been a historic, what we can say, upcomings and downs in these aspects of imbalances. ಅದನ್ನು ಇರಾಡಿಕೇಟ್ ಮಾಡುವಂಥ ಪ್ರಯತ್ನ ನಡೆದಿದೆ. But after the small districts which have been carved from these larger districts, you might have seen these administratively revenue districts have grown. I was in Gadag, I said, for four years almost. I have seen the development because Gadag was part of Dharwad district. Dharwad, Haveri and Gadag all put together, it was one district. Small districts ಮಾಡಿರೋದ್ರಿಂದ administratively, revenuely, it has helped for the development. Same thing with Ballari district. Ballari itself has been coined to Vijayanagar and Koppala now. Gulbarga has been recarved as Yadgir and Gulbarga. ಹೀಗಾಗಿ ರೆವಿನ್ಯೂ ಡಿಸ್ಟ್ರಿಕ್ಟ್ಸು ಉಳಿದಿರೋದು ಎಲ್ಲ ಪ್ರಯತ್ನ ನಡೆದಿದೆ. Administratively, it is easier when there is a small thing. Number one. Secondly, which is another important system which has to be done as a bottom-up approach, is from Gram Panchayat, instead of centralization, decentralization ಆಗುವಂಥ ಪ್ರಯತ್ನ ನಡೆದಿದೆ. ಈಗ in few departments the decentralization has already happened. And if this decentralization happens across 27 line departments, there are 27 line departments, avaga administratively also this will happen. It will take little time, but these imbalances, to an extent, the gap can be reduced over a period of time, which at least I may see for another decade.
Sir, I have one question. In regards of, you have mentioned beautifully, Karnataka has good manufacturing potential. I am from Kalburgi. I know there is limestone, there is sandstone, we are very mineral rich. So I didn't find that manufacturing sector. What is your take on that manufacturing sector?
Yes. There are three important zones of the North Karnataka region of 14 districts. The kind of cement industries which are there available in Gulbarga and nearby places, Turdal processing industries which are there, cement industries which are there, sugarcane industries which are there, and those industries, there is a manufacturing facility available, but it has to be enhanced. Invest Karnataka ಬಗ್ಗೆ ನಾನು ಹೆಚ್ಚು ಮಾತಾಡಲಿಲ್ಲ. What investments are coming in the Kalyan Karnataka region in the next few years? There is a manufacturing potential in those areas, that is in these three districts. ನಾನು ಮೈನಿಂಗ್ ಕನೆಕ್ಟ್ ಮಾಡಲಿಲ್ಲ. ಇಡೀ ರಾಜ್ಯದಲ್ಲಿ, ದೇಶದಲ್ಲಿ ಮೈನಿಂಗ್ ಬೂಮ್ ಮೈನಿಂಗ್ ಇರುವಂಥದ್ರದ್ದು ಹೆಚ್ಚು ಎಲ್ಲೇ? ಬಳ್ಳಾರಿ, ವಿಜಯನಗರ ಆ್ಯಂಡ್ ಅದರ್ ಥಿಂಗ್ಸ್. ಸೊ ಸ್ಟೀಲ್ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರಿಂಗ್ ಇನ್ ಜಿಂದಾಲ್, ಏರ್ಪೋರ್ಟೇ ಇದೆ ಅಲ್ಲಿ ಜಿಂದಾಲ್ನಲ್ಲಿ. So there is a manufacturing potential in that region also in case of mining. You come to Belgaum. Belgaum is treated as one of the important foundry cluster. There is a foundry cluster in Belgaum. There is a textile cluster in Ballari. ಇಡೀ ದೇಶದಲ್ಲಿ ಎಕ್ಸ್ಪೋರ್ಟ್ ಆಗುವಂಥ ಜೀನ್ಸ್ ಏನಿದೆ, they go from Ballari. Then you come to manufacturing sector which is in around Hubli, Dharwad. After Bangalore, the next manufacturing sector which is highest is in Hubli, Dharwad, Belgaum. So there is a manufacturing potential. More industries of small and ancillary, small and medium industries are there in northern Karnataka. SMEs ಎಷ್ಟಿದೆ, industry ಎಷ್ಟಿದೆ ಅನ್ನುವಂಥದ್ರದ್ದು. But the point is, for this manufacturing sector, the kind of connectivity, air, other connectivity infrastructure has to be improved, so that the manufacturing sector can enhance. There is potential in northern Karnataka.
ಸರ್ ನಮಸ್ತೆ. ಕೂಡ ರೀಸನ್ ಇದೆ ಅಂತಲೇ ಅನಿಸುತ್ತೆ. ಯಾಕೆಂದರೆ ನಾರ್ತ್ ಕರ್ನಾಟಕದಲ್ಲಿ ಭಾಳಷ್ಟು ನಾವು ಗಮನಿಸಿದಂತೆ, ನಾನು ಸೌತ್ ಕೆನರಾದವನು, ಬಟ್ ನಮ್ಮಲ್ಲಿ ಮುಸ್ಲಿಂ ಕಮ್ಯುನಿಟಿಯಲ್ಲಿ ಹೆಚ್ಚಿದೆ. ಹೈಯೆಸ್ಟ್ ಅವರು ಏನು ಈ ಕ್ಲೋಸ್ ರಿಲೇಟಿವ್ಸನ್ನು ಮದುವೆ ಆಗತಕ್ಕಂಥದ್ದು. ಬಟ್ ಈ ಕಡೆ ಬಂದಾಗ ಏನು ನಮ್ಮ ಮಕ್ಕಳೇ ಭಾಳಷ್ಟು ಮಂದಿ ಆಲ್ರೆಡಿ ಮಾವನ ಜೊತೆಗೆ ಮದುವೆ ಆಗತಕ್ಕಂಥದ್ದು ಈ ಕಡೆ ಇದೆ. ಸೊ ನಾನು ಅವರಿಗೆ ಜೊತೆಗೆ ಬೇರೆ ಬೇರೆ ಸಂದರ್ಭದಲ್ಲಿ ಅವರು ಕೇಳಿದಾಗ
ಬಟ್ ದ ರೀಸನ್ ಬಿಹೈಂಡ್ ಈಸ್ ಒನ್ ಈಸ್ ಏನು ಪ್ರಾಪರ್ಟಿಯನ್ನು ಉಳಿಸ್ಕೊಳ್ಬೇಕಂತಕ್ಕಂಥದ್ದು ಇರ್ಬೋದು.
ರಾಯಲ್ ಬ್ಲಡ್ ಇನ್ಸರ್ಟ್ ದೆಮ್. ಬಟ್ ಇಲ್ಲಿ ನಾರ್ತ್ ಕರ್ನಾಟಕದಲ್ಲಿ ಏನಿದೆ, ಎರಡು ರೀಸನ್ ಇದೆ. ಒಂದು ಪ್ರಾಪರ್ಟಿ ತಮ್ಮಲ್ಲೇ ಉಳಿಸ್ಕೊಳ್ಬೇಕು, ಒಂದು. ಇನ್ನೊಂದು ಏನದಲ್ಲ, ತಮ್ಮ ಮನೆ ಮಗಳನ್ನೇ ಲಗ್ನ ಆದರೆ ಶಿ ವಿಲ್ ಅಡ್ಜಸ್ಟ್ ಬೆಟರ್ ದ್ಯಾನ್ ಔಟ್ಸೈಡರ್. ದಿಸ್ ಇಸ್ ಆಲ್ಸೋ ದೇರ್ ಕಲ್ಚರಲ್. ಏನರ್ಥ, ತಮ್ಮ ಮನೆ ಮಗಳನ್ನೇ ಲಗ್ನ ಆದರೆ ಅಥವಾ ತನ್ನ ಮನೆ ಮಗನನ್ನೇ ಲಗ್ನ ಆದರೆ ದೇ ವಿಲ್ ಅಡ್ಜಸ್ಟ್ ಇನ್ ಎ ಬೆಟರ್ ಮ್ಯಾನರ್ ದ್ಯಾನ್ ಔಟ್ಸೈಡರ್ಸ್. ಇದು ಒಂದು ರೀಸನ್ನು. ಎರಡನೇದು ಪ್ರಾಪರ್ಟಿ ತಮ್ಮಲ್ಲೇ ಉಳಿಯೋದು, ಇದೊಂದು ರೀಸನ್ನು. ಇವೆರಡು ರೀಸನ್ನಿಗೆ ದೇ ಹ್ಯಾವ್ ಬೀನ್ ಮ್ಯಾರಿಯಿಂಗ್ ದೇರ್ ಓನ್ ಕ್ಲೋಸ್ ರಿಲೇಟಿವ್ಸ್ ಸಿನ್ಸ್ ಜನರೇಷನ್ಸ್. ಇಟ್ ಹ್ಯಾಸ್ ಹ್ಯಾಪನ್ಡ್ ಸಾವಿರಾರು ವರ್ಷಗಳಿಂದ. ಎಸ್ಪೆಷಲಿ ನಮ್ಮಲ್ಲಿ 3 ಟೈಪ್ ಇದೆ. ಒಂದು ಎಂಡೋಗ್ಯಾಮಿ. ಎಂಡೋಗ್ಯಾಮಿ ಅಂದರೆ ಮ್ಯಾರಿಯಿಂಗ್ ವಿದಿನ್ ದ ಸೇಮ್ ಕಮ್ಯುನಿಟಿ ಇರ್ಬೋದು, ಕಾಸ್ಟ್ ಇರ್ಬೋದು, ಯಾವುದಾದ್ರೂ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಎಂಡೋಗ್ಯಾಮಿ. ಇನ್ನೊಂದು ಎಕ್ಸೋಗ್ಯಾಮಿ. ಎಕ್ಸೋಗ್ಯಾಮಿ ಅಂದರೆ ಮ್ಯಾರಿಯಿಂಗ್ ಔಟ್ಸೈಡ್ ದಿ ಕಮ್ಯುನಿಟಿ, ಔಟ್ಸೈಡ್ ದಿ ಕ್ಲಾನ್ ಅಂದರೆ ಗೋತ್ರ. So they marry, some of the communities they marry outside the gotra. They marry inside the community, outside the gotra. And third is consanguineous marriages. I have stressed more on consanguineous marriages. Endogamous marriages are there, exogamous marriages are there, consanguineous marriages are there. These have been practiced for thousands of years in Indian population, in South Indian population. ಇದರಿಂದ our genome ಅಂದರೆ totality of all the genes in the human body, our genomes are conserved. Since our genomes are conserved, our mutations, change in the genomes, they are also conserved. Our mutations are different from European mutations. Therefore, the medicines which are produced in Europe based on their population, they are not working here. We have to produce medicines which work for our population, which are based on our genetic makeup. So that is what our institute is doing and publishing papers. This is called as ethnic genomics. Ethnic genomics is the genetic study of our own population, which will help. For example, breast cancer, if you take, the drugs which are produced for breast cancer from the West, they are not affecting on Indian population to such an extent, great extent. If you apply for 10 people, it will be affecting only 3 people. Why? Because the genes which affect the breast cancer in Europe are different from the genes which affect the breast cancer in our population. We have done this work.
I now request S.V. Hangodima to come forward and felicitate Professor Vishnukant Chatpalli, sir. Sorry, dear delegates, the dinner has been arranged in the garden area. I request all of them to head there.